drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Can you tell everyone a little bit about who you are and what your background is and
Well, I'm currently a professor in life sciences here at Arizona State University, but I've gone through quite a trajectory. I almost went to med school, then accidentally found out about this thing called graduate school and went off and studied to become a limnologist. That's a person who studies lakes, University of Minnesota, and got into ecology there. Started to learn about application of models to biology for the first time. That was good because I was interested in mathematics as well. And went through graduate school kind of rapidly, did a lot of field work, was getting a little bit uncomfortable with the generality of the results we were getting were on community structure and lakes. Then discovered population genetics in my last year. I was able to sit in on a nice course by Michael Simmons in the genetics department there. And lo and behold, then got a job at University of Illinois. I applied kind of early. I thought it might be hard for me to get a job, but I applied kind of early and before I'd written my thesis, and got a job at Illinois in the ecology, ethology, and evolution department. Was hired as an aquatic ecologist, but then was able to gradually get into population genetics with the system of organisms we were working with at the time, Daphnia, microcrustacean. This is back in the very early days of population genetics. The whole field was almost entirely theory. but there were these new things called allozymes that had been discovered that enabled us to separate proteins on gels and crudely figure out if different individuals are carrying different alleles. And it all worked the first time we applied it to Daphne. And so I rather quickly moved into this new field. My department head at the time apparently didn't pay too much attention to me and was happy for me to do any research I wanted, but I essentially shifted fields within the first 5 years. I was no longer doing limnology, but population genetics, and then just moved on to that field from there. Shortly thereafter, I moved to University of Oregon to start a new group in ecology and evolution, their first program in whole organism biology there. And I guess about a decade after that, that's when the age of genomics started. So in that transition, I was really working in the area of quantitative genetics, learned a lot of that field at Illinois from the animal breeders there. But then I was in the right place at the right time when people started sequencing genomes. And then we started seeing ways to connect evolutionary theory with genomics. Spent a while on that, wrote a book on that topic, on my ideas of how I think genomic diversity develops across the tree of life. From there, moved down to Indiana University. That was about when the book got published. And continued working along many of the same lines I'd worked on before. But a lot of people moved into the field of genomics, and I was sort of looking for things to do. And for me, the natural next step was to see if the ideas I had on genome evolution would extend to a higher level of organization. And at that time, the attraction was cell biology. And so since about I guess since around 2010 or so, I've been focused on trying to help develop this new field of evolutionary cell biology. Of course, there's a really sophisticated field of cell biology out there with exquisitely detailed measures of cellular structures and rates of reactions and so on, but it's really far removed from evolutionary biology. On the other hand, most of evolutionary biology pays no attention to what goes on inside cells. And so, to me, the natural mechanisms that are relevant to understanding evolution must start at the cellular level. That's where all phenotypes are built. And so we're now trying to bridge this gap between evolutionary theory and diversity of cells within and among phylogenetic lineages. And as I moved to Arizona State University, I started this whole new center here called Center for Mechanisms of Evolution. And that's more or less what we're focused on. We're sort of a group of misfits from different areas of biology, all focused on developing this new field.
So I want to start off by just giving people a sense for what the tree of life actually looks like. So can you tell us a little bit about what the major branches of the tree of life are and sort of how big and how diverse each one is? Like how diverse are bacteria compared to animals, multicellular organisms, and so on and so forth?
Well, the tree of life has undergone quite a transition since I started at the University of Illinois. I was there at an optimal time, actually. One of my colleagues was a guy named Carl Woese. And Carl figured out how to sequence RNAs, and in particular ribosomal RNAs. And he used that to attempt to develop the first deep branching form of a tree of life. And of course, discovered this big split between various prokaryotic lineages, in particular, one group that became known as the Archaebacteria, and another at the time was known as the Eubacteria. And so suddenly there was this two-domain of life tree. Before, all prokaryotes were just lumped into what was thought as one big phylogenetic group. Of course, this I should go back. I shouldn't call it the two-domain of life. There were two domains of prokaryotes. The third domain were us eukaryotes. There's a big question of where the eukaryotic lineage fell in the tree? Was it outside of all prokaryotes? I think that was the natural inclination initially, or was it derived from inside the bacteria or the archaea? Was it a mosaic and so on? A lot's changed since that. This was a revolutionary finding, the archaebacteria, of course, but now that we've sequencing whole genomes and get a much broader assessment than just the ribosomal RNAs, it's pretty clear that we eukaryotes are derived archaea. So now we've gone back to a two domain of life. The two domains are no longer, though, prokaryotes versus eukaryotes. It's the bacteria on the one side of what we think is the root of the tree, and on the other side are the archaea. And essentially, we eukaryotes are derived archaea. We're one branch within the archaeal lineage.
Okay,
so that's the big picture. Yeah.
So at one point, we kind of thought there's simple organisms like bacteria, and then there's complicated organisms that include us and some other things. And then eventually we figured out there's actually two sort of completely different types of simple single-celled organisms. And then there's the complex organisms, and we'll get into that a little bit in more detail later. But what we figured out, it sounds like, is that we didn't sort of evolve independently of those two. We sort of emerged, eukaryotes, complex organisms, emerged from one of those types of simple bacteria.
Yeah, that's the prevailing idea now, and it looks pretty solid. It's fair to say we're a mosaic, though, in the sense that somewhere below the root of eukaryotes, the archaeal lineage that led to us picked up endosymbiont, now called the mitochondrion. And that endosymbiont was pretty clearly derived from bacteria. We've known that for quite a long time.
I see. So one type of single-celled organism sort of absorbed another completely different type.
That's right. And many of the genes inside the mitochondria were transferred to what is now the nuclear genome of eukaryotes. So we're really a mosaic of the two major prokaryotic domains of life in some sense. There's still some debate about, the timing of events here and so on, but this seems to be the really broad picture at this point in time.
Mm-hmm. And
no pun intended.
So you've got these sort of three big branches of life, two types of prokaryotes, which I'll just call simple organisms, and then you've got the branch of complex organisms called eukaryotes. How dense are each of these branches? Are there way more bacteria and simple organisms than there are eukaryotes and complex organisms like us, or is it comparable, or how do we start to assess that?
Well, that's a good question, and it's partly unresolved. How dense? That sort of is asking how many species there are and so on. And we're getting more of a glimpse of that because we can do environmental sequencing of DNA where we don't even really look at the organism. Many prokaryotes are uncultivatable. I guess the bigger question is how deep are the branches in the different domains? And it's pretty clear now that if you just think about the depth of all the branches and diversity in the entire tree of life, most of it is in fact prokaryotic. Even within, as I said, we're just one branch in the archaebacteria, which contains many, many deeply divided lineages. So we're one branch and one side of the root of the tree of life. Most of us eukaryotes are unicellular. And so there's many deep branches of unicellular eukaryotes, as though there was a quick radiation of the eukaryotic lineages. And then a small number of us decided to go down the road of multicellularity. Metazoans, or animals, of course, are one of the more prominent ones, and land plants and other fungi to a certain extent. But us animals are a very tiny part of the branch of the entire eukaryotic tree of life, and mammals an even tinier speck on the tree. So most biologists, even evolutionary biologists, tend to study land plants and well, vertebrates. I mean, there's quite a bit of invertebrate biology as well, but we're really studying only a very small portion of the entire tree of biological diversity. Most of biological research is based on a very small fraction of the tree of life.
Hmm. And so because it's so hard to cultivate and to just find life forms that are out there, there's, probably a whole bunch of stuff we haven't discovered. Do we have any sense for how much of the tree of life we've actually discovered and how much is out there to be discovered?
Yeah, it's a real good question. Um, I mean, this whole idea of us being derived archaea has only come about in the last 10 years. And that was a consequence of sampling some, deep sea samples, just doing what's called metagenomic sequencing. So just taking a lot of DNA out of a bottle of mud and then, sequencing to a high enough depth that you can actually start to construct the organisms that are down there, even the absence of any visual data on them. But, that's just one, you know, semi unusual environment. There's almost certainly other, uh, maybe very substantial discoveries to be made. We still have uncertainty about what particular branch in the archaea eukaryotes came out of for this very reason. And we may eventually find particular lineages that are, um, intermediate states between current archaea and eukaryotes. So this is the problem we have right now is most eukaryotic lineage that we've looked at, like everything was there. So mitosis was there and internal organelles and so on. And so there's this big morphological gap between us and archaea, but it's not to say there aren't some, deep archaeal lineages with intermediate phenotypes that might then help us understand the order of events in the emergence of eukaryotes.
Hmm. Going back even deeper, when we think about, so when we look at the diversity of life that we have uncovered, and we think about the origin of life, do we know pretty definitively there was one origin of life on Earth, or what do we actually know about the origin of life? Is that something we can figure out? Is it something that we know happened once, or at least once? Is it possible that multiple origins of life happened, and then there was some kind of mixing of them? What do we know about that root of the tree of life?
Well, there's a lot of things to unpack there. Um, we think that all current lineages that we're aware of go back to a single origin. That's based, though, on a very small number of genes shared, less than 100 genes that are shared among all organisms. We need those genes they provide the information on developing genealogies of shared and unique differences. So there's not a lot to go on there. There are a number of other issues here. One is that the way we replicate DNA, archaea and us derived eukaryotes replicate DNA is using machinery quite different than bacterial lineages use. So that's led Eugene Koonin and colleagues to suggest maybe replication mechanisms in these two lineages today evolved independently. There very well could have been many other independent lineages of life that were going on at the time. There's this thing that we call LUCA, the last universal common ancestor. That's the ancestor to all of us living organisms on Earth. Could very well have been other lineages that were around at the time that went extinct. It's unlikely we're ever going to be able to resolve that because DNA, the remnants, the molecular fossils we need are just not going to be there. We can't even formally rule out the possibility that life originated on another planet and was transmitted here by a comet or so. So we've we know that based on phylogenetic information using molecular fossils and gene genealogies that seems to have started very soon after the Earth originated. Getting very, very close back to 4 billion years. So things happened pretty quickly. And of course, at that time, well, the earliest forms of life would have been pre-cellular. We're going to have no information on that. There's this whole hypothesis about an RNA world. Carl always had this idea that the earliest form of life was sort of almost a kumbaya sort of community in which you had all kinds of, became very unclear what an organism even was under his view and what a population was and so on. But then eventually cellularity took hold. And now we've got independent lineages where individual genotypes are linked inside internal cell membranes. So there's a direct connection now between genotype and phenotype and inheritance from parent to offspring cell.
Mm-hmm. What's the best estimate for when the first cell evolved?
Well, the latest estimates I was alluding to, again, this is based on shared inheritance of this small number of genes that we all have, and making assumptions about what a thing we call the molecular clock.
Mm-hmm.
So we can measure divergence among phylogenetic lineages. We can do that, there's X% divergence, but we need to know how to translate that into time. Mm-hmm. How many changes occur per nucleotide site in a genome per unit time. So we have to use these calibrated molecular clocks. The latest estimates are pushing the root of the tree of life back to about 4.1 billion years. So this is getting very tight because we think the Earth originated about 4.4 billion years.
So is it fair to say that over time, our understanding of our estimates of the origins of life have gotten closer and closer to the origins of the Earth? In other words, that life emerged more quickly than we used to think.
Yeah, these molecular estimates are pushing things back farther. There are quite convincing fossils. We don't know what they are, but we're quite convinced they're biological. Certainly go back to 3.8 billion. So that much we're sure of. But now the molecular data are pushing things back even further.
And, going back, so you said there's these about 100 genes that are sort of common to all life forms. What are those genes encoding? That's related to the question of what are sort of the universal features that all forms of cellular life share? What are the structures and the cellular functions that they're all doing that are that core set of things?
Well, the core set of biology, what is biology made of? What's the basic biology all organisms have is pretty broad. But over time, there have been what we call non-orthologous gene replacements and so on. So the same function that's in a certain lineage won't necessarily be carried out by the same gene. And there have been probably de novo origins of different genes and so on. So there are a lot of functions that we share, things like, the Krebs cycle. We all make lipids to produce membranes, glycolysis, so things that go back very deep. All the translation machinery, as I just pointed out, the replication machinery is quite different. The translation machinery, the ribosome, that goes back all the way. That's why Carl was using ribosomal RNA to really look at the entire tree of life. The ribosome itself is a bizarrely complex organelle. There depends on what lineage, but there's on the order of 100 proteins wrapped around the ribosomal RNA. Some of those proteins seem to be shared by everybody.
So this is the protein structure in cells that takes genetic material and transforms it into proteins. And
correct.
Parts of this go back all the way as far as we can tell. That's what you're saying.
Yeah. Yeah. So we really have, there's some bizarre features of biology. We have genomes made of DNA. Uh, that's where the messages are. That's where the vocabulary is to produce proteins. But the DNA has to be transcribed into these things we call messenger RNAs. That's RNA. The RNA then has to be read by the ribosomes that translates codons, which are each 3 nucleotides long. Each one stands for a particular amino acid. And then there's a stop codon. 3 of them are stop codons. 3 of the triplets are stops. Um, the ribosome works together with things called transfer RNAs that bring in an amino acid that then links to it has an anti-codon that then reads off the codon. So all biology has tRNAs and ribosomes. So those are an example of things that go all the way back.
Hmm. And I suppose that's why people have ideas about this RNA world, that RNA and proteins were there before DNA came into the mix.
Well, that's a good question. Did proteins come before DNA or vice versa? There's a strong feeling that RNAs had to have been around early on. The fact that transfer RNAs are made out of RNA and ribosomes are made out of RNA suggests that uh, there must have been some code early on. Whether that code itself, the genome itself that was being read off, was also RNA, and then there was later DNA takeover is a bit unclear. But given there was a code, then seems likely there were amino acids doing something, right? Because the code is for the readout of amino acids. Whether they were being read out to make the kinds of complex proteins that we have today is another question as well.
And so I think you hinted at this. How diverse is cellular metabolism? Are there core features of metabolism that are shared across the tree of life in terms of how we're generating ATP and making energy for the cell?
Yeah, there are certain ones. We have one would be making ATP. We have this bizarre molecular machine called ATP synthase. We all use this. By all, I mean everybody across the tree of life. It's a turbine that sits in membranes of bacteria and archaea. We pump out hydrogen. We produce hydrogen ions from organic compounds. That goes through a shared set of machinery called the electron transport chain. That also sits in membranes of prokaryotic cells. That leads to the pumping out of hydrogen ions outside the cell membrane. That produces a hydrogen ion gradient. Protons are hydrogen ions. Those protons then go back through this molecular turbine called ATP synthase, twirling it around. And in that process, ADP is converted to ATP. So this is I find this one of the most bizarre aspects of all of biology. Essentially, think about building a dam on a river for a hydroelectric plant. And, normally you would check the river to make sure there's water in it before you build the dam. And then the water just naturally comes down, goes through the turbine, and makes electricity. Well, ATP synthase is sort of like building a dam and then realizing, whoops, where there's no water above the dam.
Now you got to get the water up
So,
there.
our water in the case of ATP synthase is proton gradient, but we have to pump that out outside of bacterial cells so that it naturally flows back in through ATP synthase.
I see. Would that imply that the original cell maybe it needed to exist somewhere where such a gradient was already present in the external environment?
Yeah, that's the idea. It's one idea. It's a hint that, why are we using a proton gradient? Where did that come from? Well, maybe because the original form of life was sort of naturally exposed to some kind of proton gradient.
Hmm.
One idea is that might have happened around hydrothermal vents,
Hmm.
where geology is just naturally producing a gradient. That's a really attractive idea because now you've got a non-biological, long sustaining force producing sort of a gradient.
Hmm.
Now,
And
there's
then
a big
and then
step between that gradient and producing the molecular machine that we use today.
I see. Okay, so that would be why one idea is these deep sea vents could be a site of the origin of life,
Yeah,
cellular
that's
life.
one proposed location.
Hmm.
People
So,
are
but
liking to think about these things and just on the basis of the peculiarities of biology today. Are these remnants telling us something about where life originated? Why did
hmm.
we use certain metals in our cells? Only a handful of them and not others, and so on.
So all life uses ATP for energy. There's no, there's a lot of probably variation on top of that in terms of, obviously you've got plants using photosynthesis and, animals, making the ATP from different sources, but they're all using ATP. They're all using this turbine, this molecular machinery that relies on this proton gradient. That's a universal.
That's a universal. So that goes back to this thing we call Luca. So that would be another example of certain set of shared proteins that we all have.
Mm-hmm. So once life got started, obviously, it's there and then it starts to diversify over time. That's the process of evolution that we're going to talk about.
Yeah.
I want to start with some just basics here on some of the basic principles of evolution. So when we talk about evolution, typically people are thinking in terms of evolution by natural selection. So there's some natural diversity that's just produced intrinsically from the way that biology works, and individuals in a population are going to vary some way or another. And some of them will do better in a given environment. And that environment is sort of selecting them. And that's the process of natural selection. Can you just define for us what is natural selection? And is that the only thing that explains the diversity that we see in biology, or even the main thing?
Yeah. So you did provided a pretty good definition of natural selection. I view natural selection as one of the most powerful forces in the biological world. If you have variation, and that variation exists at the genetic level and can be inherited from parent to offspring, and it's related to differences in fitness among individuals, then natural selection will occur. The bigger question is how powerful selection is. It's not all powerful. And natural selection can only promote certain changes that are significant enough at the level of fitness differences uh, in association with the phenotypic changes. That's what natural selection can do. But many mutations have very, very tiny effects. And in certain lineages of organisms, those effects are small enough that selection can't grab onto them and push them onto the next generation. This gets into the whole idea of noise in the evolutionary process.
Hmm.
So all would work fine, and natural selection would be capable of almost anything if populations were infinite in size and everything was deterministic. But nothing in biology is deterministic. Populations are finite in size. There's a lot of noise due to the fact that a mutation arises here and whoops, another bad one arises linked right next to it. And it's no longer has a net selective advantage and so on. So there's this noise in the evolutionary process we call drift, random genetic drift. And if that level of noise is high enough so that the magnitude of fluctuations in allele gene frequencies from generation by the noise process, if that exceeds the deterministic push by natural selection, then drift wins. So there's this sort of fine point we call this the drift barrier. There's this fine point where the two forces are essentially equivalent. If you go over to one edge far enough, then selection wins. If you go over to the other edge far enough, then random genetic drift wins. And so we think that, especially at the genomic level, drift wins in many, many cases in certain phylogenetic lineages.
Hmm. So if I'm understanding you, the bigger the population is, the more individuals in a population, the easier it is for natural selection to act in the way that we normally think about it, for it to push changes, quote unquote, on purpose into the next generation because they have better fit, they're better matched to the environment. But the smaller the population, the more likely it is for this noise to dominate and for natural selection not to be able to work so well. And so I would imagine that as you sort of go from one branch of the tree of life to another, as you go from something like E. coli or simple organisms that have very, very large populations to something like us, even though there's, a few billion people on Earth, that still probably pales in comparison to the population of E. coli, swimming around out there. That would mean that this sort of drift dominates in the sort of the physically bigger organisms because they have smaller populations.
Yeah, that's a pretty good description. I mean, there's a negative relationship between the size of an organism and its population size. That's certainly true.
Mm-hmm.
And bacterial populations are literally astronomical in number. It's a little more complicated than that. Something I was alluding to is that the only determinant of the magnitude of drift is not just numbers of individuals. It's also the fact that our genes are tied together on chromosomes.
Mm.
And this causes selective interference. So if genomes could be constructed in such a way that every nucleotide were separate, then natural selection could operate on each nucleotide in a separate way. But we're all linked together on genotypes, and that causes what's called selective interference.
I see. So if I get a mutation that's in a gene on one chromosome and it makes me better at surviving in some way, but then I have another mutation on the same chromosome in a different gene and that makes me worse, those things can cancel out because I have to inherit that whole chromosome all at once.
Yeah, that's one thing that can go on. That's called selective interference. Another kind of selective interference that's very, very broad-based as far as we know, that occurs in all organisms is related to the fact that most mutations are deleterious.
Mm.
And so there's this constant rain of deleterious mutations coming down on all chromosomes and all organisms. And then those are being weeded out of the population. And when they're pulled out of the population, they pull out any linked genes with them. And so in a sense, the population size can remain really large numerically, but genetically it's behaving as though it's quite small because you have this persistent purging of variation that would otherwise be there in an physically enormous population.
Mm. So if most mutations are deleterious and you're purging those from the population, this means that inevitably you're going to be taking away new adaptations at some rate, even though they would be adaptations because they're tied to these deleterious mutations.
Yeah, in some sense, yeah. So this becomes a really powerful force. So, bacteria, like an E. coli population has what we call an effective population size, which is very different, absolute, of only about 1 billion individuals. That's a very tiny pile of E. coli cells. But genetically, the population is behaving that way in a genealogical sense. Still, that's a lot of individuals. And that allows natural selection to be quite effective. It can basically take advantage of mutations that have an effect, roughly 1 over the population size, an effect as small as 10 to the minus 9 on fitness.
What about human beings? What would do we know humanity's effective population size?
Well, there's a lot of us on the planet today, but historically, the human effective population size, you know, there's a lot of us today, but that wasn't true even 10,000 years ago.
Hmm.
And historically, the human effective size is only about 10 to the fourth individuals. And, numbers like that are common for lots of vertebrates and even some invertebrates.
So,
And
so
that
in other
means,
words, even though there's billions of people walking around on the planet today, genetically, the diversity we have looks like we're a population of tens of thousands.
yeah, that's right. New diversity generated by mutation hasn't quite caught up. We're what you would call out of equilibrium.
Mm.
Um, But the point here is that these reductions in population size, especially in multicellular organisms, mean that natural selection is going to be much more coarse grained. It can only take advantage of mutations of relatively large effect, large enough to overcome the power of drift, whereas uh, selection in bacteria can be very, very fine grained. Things can be very, very fine tuned over evolutionary time.
And so, is it so? So, can you give us? So, you said most mutations are deleterious. So, you get a genetic mutation, it can harm the organism's chance for survival. It could enhance the organism's chance for survival, or it could just be a wash. It could be neutral, somewhere in the middle. Can you give us a sense for how common each of those is? Like are adaptive mutations like very, very, very rare? Are there a lot of mutations that are sort of neutral and they don't really matter so much? Or is everything just, is 99% of everything deleterious?
Yeah, that's a good question too, that evolutionary geneticists are still struggling with. We're trying to catalog what's called the distribution fitness effects among all de novo mutations. What fraction are beneficial? What fraction are deleterious? And of the beneficials, how many are so close to, they're very, very slightly beneficial, selection can't take advantage of them. In general, we think that only a small fraction are beneficial. Maybe 1% or so of mutations are beneficial. Most are deleterious, and that's just the consequence of the eons of natural selection refining uh, structures and functions in organisms, so that most tweaks are going to make things worse.
Mm-hmm.
But things do move on when organisms are exposed to new adaptive challenges.
Mm-hmm. Well, when we look at animals and other creatures, and when we think about why they got to be the way they are, there's this natural tendency in evolutionary biology to think about things in adaptive terms. And we say, like, okay, this animal's got a really long tail, or it's got horns, or it's got this or that. We naturally want to think, okay, why did that evolve, in the sense of how are those adaptations for the organism? How did it enhance the organism's survival? But if these sort of neutral and random processes can be so strong, especially in relatively large organisms with small populations, what does that start to tell us about, like, how much of what we see in the tree of life actually evolved by natural selection and is adaptive, versus how much of it is just basically random or close to it?
Yeah, well, that's a broad issue that we're still struggling with. Um, I guess one example would be genome architecture in different lineages. So bacteria have very, very streamlined genomes. They're almost 100% coding DNA, 95% plus, very little excess DNA between genes and so on. And then you move into eukaryotes and, a lot of excess DNA in between genes. Our genes are split up by things called introns. So our coding DNA are actually islands in a sea of intronic DNA that actually gets transcribed, but then has to be spliced out for to make a productive messenger RNA by a machine called the spliceosome. Bacteria don't have these kinds of introns. They don't have this kind of problem. Our genomes, especially in animals and land plants, are just littered with what are called mobile genetic elements, or some people call them jumping genes, transposable elements. the human genome is only about 1.5% coding DNA. So how did we get here? And why is it such a mess in us multicellular lineages and in eukaryotes in general of the bacteria? The idea is it was a consequence of a reduction in effective population sizes when the eukaryotic domain started to emerge, and then further reductions in multicellular lineages. That made it easier for these genomic changes to accumulate in an effectively neutral fashion.
Hmm.
So,
so if I'm hearing you correctly, basically we when we compare simple organisms like prokaryotes to complicated organisms like eukaryotes, one of the things that what we mean by complexity in one way is genomic complexity. So we've got more stuff. There are more features to our genes. It's not just purely chunk of DNA coding protein. There's a bunch of stuff in the middle, like the introns and those non-coding elements that you mentioned. So I guess in principle, there's two ways you could start to think about that. One would be, okay, all this complexity must be doing something. It must be buying us something. It's been selected in because there's an advantage to having all that extra stuff. The other way to think about it would be that because the populations are smaller in these types of organisms, this stuff just accumulates passively because selection can't really fine tune things and get rid of it. And it sounds like you're saying that a lot of it can be explained in that way, that it's just the natural passive accumulation of stuff based on population size.
yeah, that's basically my view of all this. It's a passive expansion of excess DNA in genomes. Now, if the mutation bias was for deletions, things would go the other way, and in sufficiently small populations, you're just being pushed by mutation pressure.
Mm.
And for mobile genetic elements, they have a built-in mutation pressure. They're copies of DNA, selfish DNA, that can make offspring of themselves and insert in large chunks in other regions of the genome. So that's why we're biased towards large size. But there is an interesting point to be made here, is that we should make a distinction between the origins of alterations in something like genome architecture or any other feature of an organism, which could be by completely passive mechanisms, not advanced by adaptive mechanisms. But now you've sort of changed the genomic real estate, and you have, different kinds of material upon which natural selection might operate. It wasn't there before. A good example of this would be introns. As I said, our eukaryotic genes are in pieces, and in us multicellular species, the coding DNA are little tiny islands separated by these really large introns. And in multicellular lineages, these have become useful because it's possible to differentially splice out introns. So you can have alternatives, tissue-specific splicing of genes, and you can make different variants of genes that might work in one way in one tissue and be utilized in another way in a different tissue.
Mm-hmm.
So that's a way of thinking about how introns might be advantageous in certain lineages, in multicellular organisms with different tissues, for example. But introns evolved at the root of eukaryotes before there were multicellular organisms. So you can't invoke, tissue-specific alternative splicing as the mechanism that, promoted intron evolution by natural selection. That came later.
I see. So it sounds like basically due to the interaction between drift and selection and the population size, you can get these increases in genome size and complexity by completely passive means. It just sort of naturally accumulates over time. But in doing that, you're sort of creating a richer, more diverse substrate upon which selection can subsequently act. So you can take advantage of some of this complexity that emerges passively and now do things that you couldn't have done before it emerged.
Yeah. But you're going down a certain pathway that was, developed by non-adaptive mechanisms. Mm-hmm. That's a lot of what biology is all about, is, historical events that, set us down a certain course forever, such as ATP synthase, a bizarre way for us to produce energy, but that was the state of affairs in the last universal common ancestor, and there's no way going back anymore. Same thing with the ribosome.
Hmm. And what does this say about the reproducibility or predictability of the evolutionary process? So, for example, if you go back to the origin of life, or you go back to the origin of eukaryotes, or you go back to the origin of animals, and then you let things play forward in time again for 500 million years or whatever, would you see similar things play out, or could it be completely different from that starting point?
Well, I don't know. I'm capable of that kind of crystal gazing, but um, I mean, there's a lot of other events to worry about here because it's the Earth's geological history too that has played a role here. So it's that's a very difficult question to answer, but drift is a chance process.
Mm-hmm.
And there have been historical contingencies where, for whatever reason, certain kinds of changes arose in an ancestor to very, very large groups of organisms for some vital function that can never be lost. So, in that sense, there can be these ratchet like effects. Many of those, I think, would probably be unpredictable in certain geological contexts, not knowing the particular settings.
Mm-hmm. Um, okay, but it sounds like there's a quite a large role for pure chance here.
Yeah, well, that's what I would certainly say. Sure.
Hmm. When we think about the evolution of complexity and we think about the evolution of bigger cells, bigger genomes, going from unicellularity to multicellularity, can you give us a sense for so when we think about what a cell is doing, it's transcribing DNA, it's translating proteins, it's creating gradients of ions, it's doing all sorts of stuff. All of that stuff costs something that can, and that cost can be measured in ATP. So certain things can be done efficiently or cheaply, certain things are more expensive. Among the core functions of a cell, what are the sort of most important things energetically in terms of, I guess, the most expensive things? So what are the most energetically costly functions of the cell, and how does that start to constrain or influence the evolvability of those things?
Yeah, this is something we're just starting to work on. So we can now catalog, how much it costs to make each of the 20 amino acids, each of the four nucleotides, lipids, and so on. So we have a catalog of things like that, and then cost some energy to, glue amino acids together to make a protein and so on. So we have a pretty good sense now how to calculate the cost of making various parts of cells. And so we're trying to merge this field of what's called bioenergetics now with evolutionary cell biology to get at these very questions. Some of the things that are quite expensive are lipids. Lipids are energetically much more expensive. It takes much more ATP to make a lipid than it does to make a amino acid or a nucleotide. Well, our cell membranes are made of lipids. So, and our internal membranes in us eukaryotes are made of lipids. So we eukaryotes are investing a lot in basic biological substrate lipids that prokaryotes don't necessarily invest in. Now we use those internal organelles for a lot of different things, but it's just one example. The mitochondrial lipid membranes are very, very expensive. So we have this thing we call the powerhouse of the eukaryotic cell, the mitochondrion. That's where we make energy. That's where our ATP synthase is. So before I alluded to ATP synthase being in the cell membrane, the plasma membrane of bacterial cells, it's not in our plasma membranes. It's in our mitochondrial inner membranes. So that's where we make energy. But that little powerhouse of the cell is also pretty expensive to keep going because it's made out of a lot of lipids. And that's just sort of one example of what we've been able to start moving in this direction. Unicellular lineages swimming turns out to be a pretty high investment. It costs a lot to swim with flagella, and the flagella themselves cost a lot because they're made of long concatenations of proteins. So we're still at a very early stage of trying to figure out just what, how is the budget of a cell partitioned up. But basically, the estimates we made, I like this one quite a bit. About 1/3 of the investment of cells in its entire life history, from parent cell to baby cell, about 1/3 of the entire energy expenditure goes to making the cell membrane, and in bacteria also cell walls. If you buy a house and talk to a realtor today and ask them, how much of my budget should I be spending on, my mortgage bill per month and so on, and they'll tell you about 1/3 of your salary. So I find that quite interesting because apparently, the bugs have figured out, that rough budget apportionation themselves a long time ago.
Wow. So a substantial fraction, about 1/3 of the energy budget of a cell, is going to constructing and maintaining lipid membranes.
Yeah. Well, just the external housing. Yeah.
Interesting. When you think about like the different ways we can make ATP in our cells, so you've got like glycolysis, you've got just different ways of generating energy from different energetic substrates like dietary fats, dietary carbohydrates. Are some of those more energetically efficient than others in terms of how much ATP we can produce? Like, is it more efficient to consume lipids than sugars or something like that?
Well, an individual, if you're breaking it down, I mean, it all ends up going through things like glycolysis and this thing we call the TCA or the Krebs cycle. I mean, an individual, as I said, an individual lipid molecule is more expensive to make in terms of the ATPs required to make it than, say, a glucose molecule would be. But that also means you can squeeze, it contains more energy itself, so you can squeeze more energy out of it. So that's one way of looking at it in terms of, dietary contributions to energy that can be extracted.
Another phenomenon that's sort of interesting to me is called senescence. So cellular senescence. Can you tell everyone what that is and where it comes from?
I don't know if I can tell you that. That's one of the
well, let's just start with the concept.
What it is a gradual breakdown in the if you like, the homeostatic mechanisms of the cell. But for single celled organisms, we don't know very much about this at all. You have to follow lineages of individuals and you have to know who's mom and who's offspring, because you want to be thinking about the senescence of the maternal cell. And there's a thing called budding yeast that, thousands of biologists work on. And the baby is the bud that comes off. So you keep track of the mother that way. And, single cell yeast do seem to senesce over time. Population lives on because the babies are, it's just like us humans, the babies are, in quotes, rejuvenated. But biologic materials have to break down over time. Chance has to occur in cells. So there has to be this gradual erosion of fitness in maternal cells. Why it arises, that's a big question. Why senescence has evolved. And that's a long-standing problem. And most of the research has been focused on us big multicellular organisms. So one of the ideas in multicellular organisms is that once you've expressed most of your fitness early in life, you've had all your kids, you're beyond some point, you're not going to be having any more kids. natural selection is not going to advance any trait that influences you late in life. Any kind of deleterious trait that only expresses itself late in life will be more or less immune to natural selection.
Hmm.
So this just will naturally occur. Um, Now, there can be grand parental effects in some extreme cases of social organisms where you're expressing a little fitness late in life through your kids' kids. But, most organisms aren't engaged in that kind of grand parental care. Another hypothesis is that there's a trade-off between genes that have features on the phenotype that are really good in life that are bad late in life. And so since selections generally, it's like a compound interest problem with a bank. The faster you get your money in the bank, you make interest faster and move on and make interest off the interest. So the earlier you get your progeny into the next generation, the better. Sometimes that might come with penalties for your fitness late in life.
Hmm.
And so those are the two major hypotheses right now for the origin of aging or senescence in organisms.
Mhm. I want to ask you even a simpler question that I don't even really understand. So if we just think of a cell dividing, you've got your parent cell, you've got your daughter cell. The new cell will look fresher. It'll be rejuvenated. It won't have the broken stuff that the parent cell does. But how does that partitioning even happen? How does the process? How does the biology work such that one one of the cells of this division gets all of the fresh stuff, and the other one does not? I don't feel like I understand that.
Well, that's a big question for cell biology, and we don't really understand it fully either. I think it's not quite correct to think that all the good stuff goes into one.
Mm.
Most unicellular organisms are just reproducing by binary fission. And so there's going to be a mix. Uh, and then it becomes very unclear, who's parent and offspring. I mean, I guess the inclination would be to call the one who's got all the good, more of the goodies than the other one would be the parent. But, that's just sort of a semantic issue here. But we think it's, things that reproduce by normal binary fission, just, a split down the middle. Um, it's not like budding yeast where there's a distinction between mother and offspring cell.
Mhm.
And then, there's lots of big questions like, I've got a molecular machine like ATP synthase. Well, does the whole complex get inherited intact from parent to offspring, or the components come apart and they're reassembled, a mixture with new and old parts at the same time? Those are difficult questions that people are still, struggling with in cell biology, but they're at the heart of the problem, I think, for cellular senescence.
Hmm. Another thing I want to ask you about is epigenetics. So if we go back even before Darwin, right? Before Darwin and Wallace came up with the theory of natural selection, had people like famously Lamarck had his theory of acquired characteristics that, you could obtain as an adult organism some new trait during your life and you could pass it on to your offspring. So that would be like, I don't know, lifting weights as an adult, getting bigger muscles, and then your baby has bigger muscles, even though you weren't born that way, you became that way during your life. It seems that recently, in recent years, people are finding examples of the inheritance of acquired characteristics. So, for example, I know that there are certain metabolic phenotypes that, say, a rodent can acquire as an adult and then pass on to their children for some number of generations. The subsequent generations, for 3 or 4 generations at least, will have some metabolic phenotype that their parent acquired during their lifetime. So it's not strictly encoded in the genes. It's some kind of epigenetic mechanism. How do you start to think about this? Are there true epigenetic inheritance mechanisms that don't depend on the actual DNA sequence? Is it truly possible for phenotypes to evolve within the lifetime of an organism and then persist in a population over time?
Yeah, that's a good question. I think there's a lot of misunderstanding about that. So I think we're all convinced now they're epigenetic mechanisms. So these are, effects determined by the cellular environment that modifies uh, various aspects of the genetic machinery, including the genome itself, but in non-perfectly heritable ways. But they can influence what happens from generation to generation. That means that natural selection can take advantage of some epigenetic effects. So you could have two individuals, for example, with identical genomes, but one's been slightly modified by some kind of methylation or something that another one hasn't, and will have more success. Um, but this is not likely to be a persistent evolutionary change. So the way I like to think of it is an epigenetic effect is a transient mark on the DNA. And that in some cases can be passed on to the next generation, but there's also a certain probability it's going to be lost by chance or just erased by natural cellular processes. The actual DNA doesn't change like that. The only way the DNA is going to change is by a real mutation at the nucleotide level. So you can have these epigenetic effects. They can be transmitted, but then there's a bit of a loss each generation.
So
And
it
then
sounds like
maybe
they're
you push.
probably inherently less stable because they can be taken off and removed through normal cellular processes like
Yeah,
methylation
so
and methylation.
you could have selection persistently pushing you in a certain direction, gain a little bit of loss, gain a little bit of slippage, and you will actually reach an equilibrium between the gains and the losses.
Mm.
And selection, if it was persistent, it could keep you over at this point without any change at the DNA level. But then if you stop selection in that directional way, you will just revert to where you were before. You'll go back to the prior equilibrium level of these epigenetic effects. So it can lead to a transient change for sure, but not a permanent change by itself.
Mm-hmm. Oftentimes when we think about natural selection, we think about a population boom. So there's sort of an excess of individuals produced, and then the population is culled because not all of them can be, not all of them fit into the environment. The external environment can only has a certain carrying capacity, as an ecologist would say. There's also processes like that happen within organisms, within cells. So, for example, genes can duplicate and then their functions can sort of deteriorate in complementary ways, and you can get some complexity evolving that way. Can you discuss that a little bit about, within an individual, within one genome, this process by which there's an expansion of stuff, a duplication of a gene or a set of genes or even a whole genome, and then there's sort of degenerative processes that where you can have a conservation of the overall function, but then you get a change in the architecture for how that's actually happening.
Yeah, well, you mentioned ecology. I kind of like to think about, gene duplication as a process that's similar to, birth and death of individuals in a population. So all genes in all organisms are subject to being duplicated. Now it's not always a complete clean duplication of the entire gene, but for purposes here, let's just think about it that way. So all genes are giving birth to baby genes that are direct copies of themselves. And we discovered quite a few years ago now that this occurs at a pretty high clip in all organisms. But then what happens to the duplicate? If it's completely redundant, it's eventually one of the copies is going to suffer from degenerative mutations and be lost and eventually become what's called a pseudogene and maybe eventually be deleted from the genome. So there can be this steady state process in cells. So in a population of individuals, if you keep the age specific birth rates and death rates constant, you will reach what's called a stable age distribution. That stable age distribution actually can tell you something about the birth and the death rate. So a rapidly growing population will have lots of juveniles in it and fewer adults. And demographers use that information to infer past history of birth and death. And we can do that with duplicate genes and genomes. So in bacteria, for example, I mentioned before, they're very streamlined genomes. They duplicate genes pretty much the same rate that we eukaryotes do. But they have very few duplicate copies of genes in the genomes, probably because an additional copy is usually deleterious. And so the death rate for the excess copies is relatively high. Whereas in us multicellular species, we have enormous numbers of duplicate genes in the genomes, in all of our genomes. And many of them are very, very young, but many of them eventually make it to an old age. Some get lucky and discover a new function. Sometimes they just partition up old functions. So you had an ancestral gene with multiple functions and you duplicate it, and now they partitioned up different functions, say in different cell types. And so we call that subfunctionalization. So I like to think of this whole process as very similar to the way population ecologists think about the demography of individuals.
And how do you start to think about things like the speed of evolution in a given lineage? Obviously, that's going to vary for an E. coli compared to a creature like us. But when you think about, right, that there's a certain fundamental limit on the rate of mutations. There's this, intrinsic error rate of the DNA replication machinery. There's, how many mutagens are in the environment that the organism is exposed to. And then there's the population size effects that we talked about that dictate, how easily certain mutations can spread through the population. How do we start to think about the limits on how quickly adaptations can happen, how quickly the genome can change?
Yeah, well, that too is a really good question. There's several ingredients here. One is, we need to know something about the mutation rate. 30 years ago, we knew almost nothing about rates of mutations in different lineages, and today we know a lot about it. And rates are per nucleotide per generation are, 100 to 1,000-fold higher in eukaryotes than they are in bacteria. So that might seem good, but then you got to take into account the generation time. Bacteria might have a generation of only, half an hour or an hour, whereas some of us big multicellular organisms, it's more on the order of 10, 20, 30 years and so on. So you got the element of time working. We have bigger genomes, on the other hand. So how does that all factor in? There's more nucleotides that might contribute to adaptation. On the other hand, we just have smaller effective population sizes, so natural selection can't be as effective. So all these things work together to define the limiting rate of evolution. But we only have a crude idea how all this really works. The reason is because we can kind of talk about this thing at the level of the nucleotide, but we don't yet have a good understanding of how nucleotide variation translates into phenotypic variation on which natural selection can operate. This is a really important question today in a changing world, because we'd like to know, what is the limiting rate of environmental change beyond which organisms just cannot adapt.
That's probably especially relevant to humans.
Global change. Maybe they're even keeping up. Well, not keeping up. What we think goes on is, in the perfect world for an organism, say environmental change is changing on a gradient like this. What will happen with the organism will be chasing that environmental optimum. But there'll always be a lag, because every time you move up, the environment moves up as well. So there'll be this lag, and this ultimately leads to what we call a lag load. You're evolving, in response to the changing environment. But as the lag load gets bigger, your population size is declining. And if your population size is declining, that means you're going extinct. You cannot keep up.
Hmm.
So there's a critical rate of environmental change beyond which adaptation must not be possible, must not be sustainable.
Hmm.
And we don't have definitive answers on what that is and how that varies among phylogenetic lineages.
Hmm. Yeah, that's interesting because, well, it's interesting as a human being because, I think you could make the case that our environment changes faster than any other organism's environment because of our cultural capabilities.
Yeah, but we modify our environment and we live in ways. I mean, that's what modern medicine's all about, right? We live in ways that protect us, not against just deleterious environments, but deleterious mutations.
Mm.
So, the way I think of it is a lot of natural selection on humans is relaxed as just as a natural consequence of modern medicine. That's what modern medicine's all about, living with our genetic afflictions.
Hmm. Yeah. So, I mean, not only do we have a small population size because we're big creatures, relative to smaller creatures, but because humans, because we have the brains we do and we can invent things like we do, that's a further relaxation of selection because we don't, we're not subject to the same survival constraints that our ancestors were. How do you think that? I mean, are humans effectively are would you say that we're not evolving as quickly or not adapting as much that because selection is just such a weaker force for us? Like, how do you think about human evolution and the ways it might be sort of on a different trajectory than other creatures?
Well, there's a lot of interesting questions here that, can be a bit of a landmine. Um, but as I said, modern medical procedures are all about, allowing us to live with our genetic afflictions. I mean, there's no question about that. There's some other interesting aspects of just human biology. One way of thinking about this is the so-called middle class neighborhood syndrome.
Hmm.
If the way natural selection operates is by variance in family size. And so an interesting question here is how has that changed over evolutionary time? If everybody decided to just have two kids, well, everybody's got the same fitness, right? And that would be a different form of relaxation of selection. I mean, there has to be, I'm not arguing that natural selection is not operating in some effective ways on the human population. We're, just indirectly by environmental change. Some of
Mm-hmm.
us are built better for certain environments than others. And as a whole population, this is probably having some effect. But evolution by natural selection in humans is very, very different than it is in other organisms for the mere fact that we construct our own environments.
Mm-hmm. Does that mean that just the level of influence of stochastic processes like genetic drift must be higher in our species than it would be for other species?
Yeah, I don't know that I would so much call it a issue with drift is just a relaxation of selection in some cases. I mean, there's a lot of other things to think about here. We're not one giant panmictic population across the earth, randomly mating across the earth. So there are different segments of the human population in different environments, exposed to different factors, um, exposed to different, privileges that we have with respect to medical procedures and so on. So it's a very heterogeneous sort of thing going on in the human population. And
Hmm.
there's a lot of us today, a lot more than there were historically. The 10,000 that I talked about, our historical effective size, that's not our effective population size today.
I see it's larger today.
Yeah, I mean, just based on numbers alone.
Mm-hmm.
Yeah.
What are some of the things? what do you think are some of the biggest misunderstandings that people, ordinary people, or even biologists have about evolution, in terms of things that you feel like we have a good handle on and have a relatively solid understanding of, that people are often mistaken or misled about in terms of how evolution actually happens?
Well, I think a big unfortunate aspect of biology is Darwin and some of his bulldogs over the years who've written popular books have espoused on the power of natural selection so much that for most biologists, even many well-trained biologists, even many people who call themselves evolutionary biologists, feel that everything is explainable by natural selection. And that just can't be true. But those are the avenues we've gone down. As a consequence, there's lots and lots of excellent comparative biology being done in fields that by people who don't normally think about evolution, but there'll always be some teasing paragraph at the end of the paper explaining why these differences among organisms, what must have led to this divergence by natural selection. I mean, it's just accepted this has to be the process that led to the diversification. And so it really diminishes our ability to think about evolution when we've already pre-decided that the mechanism must be selection.
Hmm. What do you think are some of, in terms of the tree of life and how it looks and how we understand it, and even going back to where life came, how it originated in the beginning, what do you think are some of the major questions related to the origins of life and related to our understanding of evolution, the process of evolution, that you think will probably start to answer in the next 5, 10, 15 years that we don't have a good handle on today?
Well, it's likely we'll have a bigger understanding of the tree of life itself. I think almost certainly there's going to be a lot of interesting lineages, microbial lineages in particular, to discover that might provide us with better insight into the order of events in the emergence of eukaryotes. The big problem with eukaryotes is, as I said, that there's many, many shared characteristics that all us eukaryotes have that must have been in the ancestor to us all, at least of the lineage we see today. The nuclear, our DNA is inside a nucleus. We have mitochondria, and the list goes on and on and on. The problem we have in trying to understand the origin of eukaryotes is we don't have any ancestral lineages that have, one or two of these changes that others don't have. So we have no way really of convincingly come up with an order of events that led to eukaryotes. This is why I'm wondering if someday we'll discover some, what we might still call archaeal lineages that have more eukaryotic-like features.
Mm-hmm.
So I would be extremely surprised if there aren't a lot of discoveries that will help us decipher the order of events towards what we call eukaryogenesis over the next few years.
Mm-hmm. What are some of the questions that you're working on today that you're really excited about?
Well, as I said, we're interested in this broad connection between cell biology and evolutionary biology. And we have a lot of unsolved problems, even theory that hasn't yet been developed to try and bridge this gap. We don't really have a full grasp of how to link the power of cell biology and amazing level of observation with evolutionary theory. And this is going to have to involve much more complex theory than just saying oh, there's two nucleotides at this one site in the genome, and let's figure out if you go this route or the other route. These are complex traits. But one of the things that we're most interested in is trying to understand how some complexities of the cell have evolved by semi-neutral mechanisms. I'm trying to extend these ideas that I've mentioned earlier about the origin of genome complexity, the origins of cellular complexity. One good example would be the ribosome. All organisms have ribosomes, but in eukaryotic lineages that we think have much smaller effective population sizes, there's many, many more proteins involved in the ribosome than there are in bacteria. There's no evidence that our ribosomes are any better at translating, more accurately, more rapidly. But somehow we picked up this excess complexity over time. And you can talk about that with a number of different aspects of cell biology. So again, this raises the whole issue, not just at the genomic level, but now at the level of cellular complexity, that, we went down certain kinds of evolutionary paths with certain kinds of population genetic environments that just predisposed us being, the ancestral eukaryotes, to go down this route of the passive emergence of cellular complexity. So that's, I think there's real possibility to broadening the field that way. And then the next step, a big step for me, will be trying to link, the things we've learned at the genomic level, now extending them to the cellular level, and then bringing it up to the level of multicellularity, and how many aspects of the features of multicellular organisms are actually generated by semi-neutral processes. And then put us in a new kind of organismal real estate upon which natural selection operates. So again, I think the key thing is separating out what mechanisms led to the origin of a certain change, and then natural selection's subsequent ability to move things around.
Well, Professor Lynch, is there anything that you want to reiterate or any final thoughts you want to leave people with about evolution or about any of the topics that we covered today?
Well, I don't think too much more. I guess my main point would simply be that, there's a lot more to evolution than just natural selection. And if we really want to understand the mechanism of evolution, we need to know how to put all these different themes together in a general cohesive framework.
All right. Well, Professor Michael Lynch, thank you for your time.
Thank you very much.
This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.